Okay, so we're going to go over some genetics and the structure of DNA and a little bit about replication as well. So we need first to just do a simple dominance Punnett square. So this is just for one gene, and this is the trait of coat color. So for this gene, it's going to be like the B gene. And the capital B is the dominant allele. The lowercase b is the recessive allele. So we want to cross two monohybrid cross, meaning one gene, and we want to cross two heterozygous parents, meaning they each have one dominant allele, one recessive allele. So when we draw a Punnett square, I do it this way. You take each parent, so it's going to be parent one is heterozygous, the other parent is also heterozygous. Take each parent, separate their alleles, and put one allele for each box. And then we'll put this parent over here. Then we cross and find out how many possible combinations these two parents' babies could be. Okay, so there's the Punnett square. Let's answer these questions. The genotype of a white rabbit, the only way that you can be white is by being two recessive alleles, which is called homozygous recessive. If you're homozygous dominant, that'd be big B, and that means you're going to be brown. If you are heterozygous, you are one dominant allele, one recessive allele, looks like that, and you're going to also be brown. So then E is just going back to the fact that if you're brown and all you know is that the rabbit's brown, you don't know if they're homozygous dominant like that or if they are heterozygous like that. Just based on their phenotype alone, you're not sure. Okay, so if we talk about PTC is just a common simple dominance thing in humans. You get a little strip of paper, and some people can taste it. It tastes bitter, and some people just put it in their mouth and they don't taste anything. They're just like, why is there a piece of paper in my mouth? This is weird. So you can either be a taster or not a taster. So if the ability to taste is the dominant allele. So if you are homozygous dominant, you taste. If you're heterozygous, you can taste. You're a taster. If you are homozygous recessive, you are not a taster. So this question is, suppose two heterozygous tasters have a large family. So when you want to predict the proportion of their children's phenotypes, you do Punnett square. Luckily, we're still on one gene here, so we're just doing a monohybrid. Yep, because we're doing two heterozygous. So we're doing that by this. So you take the first parent, segregate their alleles, put one on each box. Second parent, do the same down the side, cross them. Okay. So the proportion of their children who will be tasters. This one's going to be, so there are 1, 2, 3 out of 4 total predicted to be tasters. Okay, so that's also 75%. Or you could say a 3-to-1 ratio of taster to non-taster. Okay, so some questions. What's the likelihood that their first child will be a taster? Okay, that's going to be 75% that their child will be a taster. For their fourth child, this is a pretty common trick. It's just the same. Every single time you combine this parent with this parent, you're always going to have this same chance of same combination of possible zygotes that are built based on these sperms and eggs. So think of it like flipping a coin. Just because you got 3 heads in a row doesn't mean your next one is definitely going to be a tail. You still have a 50-50 shot every time. It's the same here. So it doesn't matter if it's their 4th child, their 8th child, 11th child, it's always going to be a 75% chance that their kid is going to be a taster and a 25% chance that it isn't going to be a taster. So that's simple dominance. We're going to move on to the next question, which is also simple dominance, but we are looking at 2 genes at once. So we're looking at the coat color trait, which is the B gene. We're also looking at the pattern trait, which is the S gene. So we know that black is dominant over brown, which is recessive. Solid is dominant over the recessive spotted. So we're going to assume that they assort independently as opposed to being linked, which an example of something that's linked would be like red hair and freckles. They normally go together, but in this case we're going to assume that hair color and whether you are spotted or not doesn't go together normally. They're completely independent of one another. So what we do here is to predict is we make a giant Punnett square, but first we have to think about each parent. So we know that we're going to cross 2 heterozygous parents. So here are their genotypes, and I'm just going to write it a little bit bigger for the video. So we know that we have this parent, and let's just think about this one parent. The first thing that we want to do to fill in the top or the side of the box is think about what is this parent's gametes, what are the possibilities of this parent's gametes going to be. So we know that when this parent is going to make a sperm or an egg, it's got to have one allele for the B gene, and it also has to have one allele for the S gene. So what we do is we think, how many possible combinations of B and S can we make? So we know from this one parent, we can make a gamete that is the dominant allele for the B gene and the dominant allele for the S gene. We can also make a gamete that is dominant B, recessive S. We can also make a gamete that's recessive B, dominant S, and recessive B, recessive S. Okay, so now we just got the top of our Punnett square here. And we do the same thing with the other parent, and this is easy because they're the same. So that's going to be big B, big S, big B, little s, little b, big S, little b, little s. Okay, so then we cross, and when we're thinking about these as zygotes or little babies, we know that we want them to go back to having 2 alleles per each gene, one from mom, one from dad. So we're gonna cross the 2 B's. So this child is homozygous dominant for the B gene and homozygous dominant for the S gene. This one, homozygous dominant for B, but heterozygous for S. Heterozygous for B, this is a big S, okay. Homozygous dominant for S, okay. Heterozygous for B, heterozygous for S. I'm just gonna finish this off. Trying to make my S's as dramatic as possible. Heterozygous for B here. Homozygous recessive for S. That's new, I think. Oh no, we did it over here. These are 2 big S's. Heterozygous for B. Heterozygous for S. Homozygous recessive. Homozygous dominant. Homozygous recessive and heterozygous. Heterozygous, heterozygous. Heterozygous, homozygous recessive. Homozygous recessive, heterozygous. And homozygous recessive, homozygous recessive. Whew. Okay, so that's that. And now we need to go back to the question What would be the phenotypic and genotypic ratios for the F1 generation? Okay, so F1 generation just means these are generation 1 after you've crossed. So what you want to do is write out what are the possible phenotypes. So possible phenotypes are, this little baby right here is black, and it is dominant for S, which is solid. So black solid. We've got how many of those? This is black and solid. Black and solid. That's 1, 2, 3, 4, 5. Not this one. 6. No. 7, 8, 9. Okay, so we got 9 for black solid. The next phenotype would be, these are all black and solid, black and solid, black and spotted. So black spot would be, okay, solid, 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 solid. This one, 1, 2, 3. Okay, so the next after that is going to be brown and solid. So let's count up how many brown solids we have. So we're looking for brown. Here's one, brown and solid. Yes, 1, 2, 3. And then the last phenotype we're gonna have is brown with spots. So we're looking for homozygous recessive and homozygous recessive, and there's only one little guy right here who's going to be brown and spots. So that's going to be our genotypic ratios. Then if the question asks you about percent, you do the same thing as we did up here. Just think about what is the percent out of 16. Okay. And that's the dihybrid cross. Okay, so for DNA replication, there were a few different ideas before they were able to figure out exactly how DNA replication happens. Now we know that DNA replication is semi-conservative, but we didn't know that right off the bat. So we did some, this really nice experiment to show, to find out like which one it is. So let's go over the 3 models first. Where should I draw? Okay, let's assume up here at the top that, here's what happens. We got the original strand of DNA that looks like this. This is our original. Okay, if the DNA is replicated by conservative manner, then that means when you replicate, the original strand comes back together and the new strand is going to be all new nucleotides. So I'll draw those in red. If we assume semi-conservative, then let me write, this would be conservative. If we assume semi-conservative, then that's when the DNA strand opens up. So it looks like this, both sides open up, and then we add new nucleotides in on the inside. Okay, and that's what we know is right now. But third idea is this one called dispersive. And that's where your new strands are sort of a mix of new and old, what do you call them? Nucleotides. Okay, so it would look, both strands would look something like this. When they replicate it. So you still end up with 2 strands, but some of it's old and some of it's new within the strand. This one you end up with 2 strands, half of it's old, half of it's new for each strand. This one you still end up with 2 strands, but one of it is completely old, one of it's completely new. So how we tested that is we grew the original in this nitrogen, like gave it this nitrogen that is this 15N. That just means the nitrogen that's, we know, so back up a little bit, we know that DNA is made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and a few other, no. Okay, so this nitrogen that's in the nucleotides is the version that has 15 neutrons. So this is the isotope of nitrogen. So we're growing it in 15, which is just bigger than this one that we're going to use later, which is grown with the nitrogen that has 14 neutrons. So this one's just physically bigger than this one. So it just, the DNA itself is just a little heavier because every single nucleotide has this fatter nitrogen than this one over here that's smaller because it has less neutrons. So again, remember that we can't like put a video camera in a nucleus and watch the DNA replicate. So we have to, this one is nice because we separate it out by weight. So what happens is you let the original one grow, and it's a big fat one, heavy one. Then the new stuff, when it's time to replicate, the only nitrogen available at that point is this light nitrogen. So anything that's made next will be with this light nitrogen. So that's how we can see. So we kind of put it all into a test tube, shake it up. So we have at the end, these will represent our test tubes here. What we're looking at here, we're going to have one separation here that's completely heavy. So it's going to sink to the bottom. Okay, and that'll be a heavy strand. Then we have this one strand, this 1/2 of our solution that is completely light. So it's going to settle up here in a light band. Okay, and think about this like oil and vinegar. Like it's just, you can see it suspended in the solution like, oh, here's this stuff and here's this stuff. And all the rest of it's kind of like water, and it's floating here and here. So that's what you see. In semi-conservative, when we look up here, both of these are the same. They're half and half. So they're not going to settle all the way down here or all the way up here. Where they're going to settle is right in the middle. Okay, so just to keep this red and black thing going, I'll do this. But you can't really see. All that you see is just these lines in the real experiment. So this is one medium band that ends up doing, because both strands are the same. So they're going to settle at the same point because they weigh the same. That's the same result that you get with the dispersive, because both of these strands are the same. They're both mixed up. They're both half heavy, half light. So that's, again, going to give us one medium band. So right now, since the results were one medium band, we know it's either semi-conservative or dispersive. So we can rule out conservative from here, but to determine between these two, we have to keep going to the next generation. So if we continue with this picture, okay, how can I do this? Let me rip this piece of paper. I can do something like this. Does that work? Oh, almost. Yeah, okay. So at this point, if we let it replicate again, and still the new nucleotides that we'll be making are also going to be light, because we're still growing them with the 14 nitrogen, then if we still are going with the conservative model, that means the original here will stay together. Okay. This red original will also stay together. But then we'll just make a copy of each, but it's still going to be made with light nitrogen, so it'll be red in weight. Okay, semi-conservative model. Ooh, this is going to get a little crowded. Semi-conservative model means that both of these are going to split open. So I'm going to draw the original black here and the original red out here. And we know that they will fill in with new red. Okay, then we're going to split this one as well. That's the original, and this original. It's also going to fill in with new red. Okay, so that's semi-conservative. If we do the dispersive model, we're going to end up with, again, we're going to make a copy of this one, and it's going to be half red, half black. That's a terrible drawing. Live probably. Okay. And this one, then it's going to make another copy, and it's also going to be half red and half black. Okay. This one's going to make two copies of itself, half red, half black. This is half 14 and half 15 nitrogen. I should be reminding us that that's what we're talking about. Okay, there we go. Whew, okay, so now looking at the second generation, thinking about their bands, we're going to have, on the conservative model, again, one completely heavy band here. Okay, sorry, so my phone died. We're going to end up with, on generation 2 in our test tube, for the conservative model we have here, we're going to see one completely heavy bit, which will all land up down here, and then 3 light bits, which will settle up here at the top. So the semi-conservative second generation, we have these two on the ends, which are half heavy, half light. So those will settle in the middle of the test tube and one medium band. These on the inside are completely light, so they will settle at the top in their own band. The dispersive, it will make 4 bands, 4 new DNA strands that are all mixed up. So they're all half heavy and half light. So that means they will still just make one medium band going across the middle. So that's how you can finally tell the difference between semi-conservative and dispersive. And again, since these are our results here, we know that it's a semi-conservative model. Okay, so moving on to this question about DNA. Let's see if I can zoom in a little bit. No, that's bad. Okay, what we want here is the function of the proteins involved. So we're talking about DNA replication here. It looks very complicated, but it's not that bad once you put it into a whole story. So we need to label all these things and talk about all their functions. So first of all, remember that in a nucleus, each of your cells, you have approximately, if we lay our DNA out like end to end, not all crumpled, like spoiled up, then that means we end up with about 2 meters of DNA in each of our cells. So that's crazy. And in each of our cells. So that means our DNA is very tightly packed up like this most of the time. So our first enzyme that's important here is topoisomerase, and this one's going to relax our DNA. So it's going to not supercoil it, unsupercoil it. And so that's puts it into a straight line here so that we can come and unzip it and do the work. Well, the next enzyme that comes in is helicase. That's here. Notice that it's going in this direction here. Helicase is unzipping the strands. So it's breaking the hydrogen bonds that are between each of the nucleotides. Okay. So since it just broke those hydrogen bonds, they want to come together really bad. Like they would rather be together. So you have to have these single strand binding proteins, also sometimes just called SSB. These stabilize the single stranded DNA. So you have to have that so it'll stabilize it so it doesn't come back together. The next thing that comes in, so now we're ready to add on nucleotides to each end of this. So what's going to happen is primase comes in. That's here on the picture. And this makes the RNA primer, which is right here, this little prime bit of RNA that comes in, and it first adds and gets the DNA replication started. So notice that we're going from 5' to 3' direction. So that means here's our 5' end, and that means this is the 3' end. That means on this strand, we've got 3' here, 5' here. Okay. Because they're complementary, they line up in kind of opposite like that. So once the RNA primer is there, DNA polymerase 2 comes in right here. It's not on your picture. DNA polymerase 2 comes in, and what it does is it adds DNA nucleotides. So it makes the strand. Okay, so it's going in the 5' to 3' direction always. So it'll land right here next to the RNA primer, and then it'll just it'll start making those nucleotides here to fill in the other side of the DNA. And it again moves from 5' to 3'. At the end, something special comes in. It comes in right here. That would be DNA polymerase 1. And it replaces this little chunk of RNA, the RNA primer, with DNA nucleotides, because our DNA isn't like little chunks of RNA along the way. So it comes in and replaces it with DNA. Okay, so that's happening. That's a very easy process. Helicase keeps unzipping this way. The topoisomerase keeps straightening out the DNA this way. It's very nice. This is our leading strand. Very simple and easy. Okay. Now, on this strand, our lagging strand, it's not quite that simple. We have the same process that happens here. We have the primase that comes in. Boop. It's going to sit here. And then the little RNA primer is going to be built in. And then we have the DNA polymerase 2 that'll come in and start adding nucleotides. Remember that it has to move in the 5' to 3' direction. So that means DNA polymerase is going to move this way on the lagging strand. And that's great. It's adding nucleotides. Great, great, great. Perfect. But then our DNA polymerase is sitting here. Our helicase is still moving this way. So now DNA polymerase is over here trying to move in this direction. Helicase has moved this way and is unzipping the DNA over here. So what has to happen is this has to let go, and then imagine helicase is now here. This is now unzipped. The DNA polymerase, the primer, everything has to come back over here to now let's say now it looks like this. Let's leave this. Here was the helicase, and now let's imagine that it has unzipped all the way to here. So this is our helicase. It has moved this way. So for our leading strand, the DNA polymerase is sitting here. Let me do this just to finish the picture off. DNA polymerase is sitting here. It can still be moving from the 5' direction to the 3' direction. You can just keep following helicase. It's great, doing great. That's the leading strand. It's super easy. Lagging strand, remember where we were when the helicase had unzipped here. We started here with the polymerase. It's adding the nucleotides, and now our enzyme sitting here trying to go this direction. It can't, because that's the end of the DNA. So it has to start again back here. So we get the primer added, we put the polymerase in. Now the polymerase can move this way from the 5' to the 3' direction. What this is making, so we keep thinking about the helicase keeps moving this way, we keep adding more this way and this way, as opposed to the leading strand, which can just keep following helicase. That's why this one's called the lagging strand. This one takes a little bit longer, okay? And what we have built here are a bunch of these special Okazaki fragments, because we've had to keep going, starting over and moving forward a little bit, going back to where the helicase is and starting again. So the second to last enzyme that you need to know, where would be a good place to put that? Let's put it over here, sure. Would be DNA ligase, which this glues together the Okazaki fragments. Okay, ligase, this lig root means glue or put together. Think of it like a ligament holds together your bones. So that's gluing together the Okazaki fragments. We're almost finished. The next thing that needs, so when we're unzipped and the helicase is moving, as soon as those SSBs leave, that's not an issue anymore, because we finish off these DNA strands. So now we're 2 strands. As soon as the topoisomerase leaves, then we need another enzyme called gyrase to come in, which does the opposite of topoisomerase. And this one, here's the definition. It introduces supercoiling again. So the topoisomerase uncoiled it into a straight line. The gyrase comes in, coils it back up. Okay, so that's the story. This looks crazy now, but that's the story. So you go from, let's move this up here. So you have your topoisomerase, which uncoils our DNA. Then you have the SSB that stabilizes. Oh, you know what? In between there, we have the helicase, which unzips. Then we have the SSB, which stabilizes the single-stranded. Because that's not stable. Because of the helicase unzipped it, the SSBs stabilize it, leave it open, when normally it wouldn't want to be open, it would want to close back up. From there, we have the primase that comes in, which makes the RNA primer. Then the RNA primer is a little chunk of DNA, chunk of RNA that primes where the DNA polymerase should show up. Okay, from there, we have the DNA polymerase 2, which comes in and it adds DNA nucleotides. Then we have the DNA polymerase 1, which replaces that RNA primer with DNA. Then remember that we have this leading lagging strand issue. So we need DNA ligase to come in and glue together the Okazaki fragments. Oak frag is what I wrote there. Then, lastly, we have the gyrase, which comes in and recoils the DNA. Okay, so that is DNA replication in a nutshell. Okay, so number 3, why is DNA replication exergonic process? This is a fun question. This is actually pretty simple. Brings back some old topics too. So we originally kind of talked about nucleotides look something like this. They have the sugar, your phosphate group, and your base. So this would be a nucleotide. What's actually floating around in your cells that comes in with the DNA polymerase is something that looks like this. Ooh. Okay. This is called a DNTP, or a deoxynucleotriphosphate. So that triphosphate should sound familiar to you, like ATP, okay? And remember from that what we know is that these phosphate-phosphate bonds hold a lot of energy, okay? And notice that in order to go from a DNTP to a nucleotide, we need to break off 2 of those. So when we have to go from a DNTP to a nucleotide, we have to break off these phosphates, which releases so much energy. That energy comes out that will be overpowering of all the building of the big molecule that we have to do. So it ends up being exergonic.